Okay, I know for some of you, you've already looked at Alzheimer's disease. One of our guest speakers that's coming up very soon works on Parkinson's, and she'll give you a slightly different take on Parkinson's than we had earlier on. But I didn't want to recapitulate something that I'd done before. And I know many of you are interested in things like Alzheimer's disease, and as a result, I wanted to go over another neurodegenerative condition that in many ways resembles Parkinson's disease, but in many ways is different. Different areas of the brain are affected, different classes of neurons are affected. The way in which they die are not alpha-synuclein dependent, they're more dependent on other proteins that we'll be talking about today. But the overall idea is that there are a number of neurodegenerative conditions, some of them affecting movement, some of them affecting mood, some of them affecting things like cognition. And today we'll talk about Alzheimer's. The other theme that we've had throughout this course is not only is neuroscience something that is innovative. So we've talked a lot about Nobel Prize laureates, and we are going to come back to Nobel Prize laureates very soon. But in addition to Nobel Prizes and everything else and innovation in neuroscience, the other reason that I want you to be really interested in neuroscience is because it should be personal. So neuroscience, if it's just static, if it's just something that you're coming and learning about because, well, maybe it's a little bit fascinating to you, maybe you've heard neuroscience is an easy discipline, maybe it's something that meshes well with psychology, that's okay. Like, you have a lot of reasons for taking the majors that you are. But beyond that, I also want you to be thinking about neuroscience to have some impact on your own life. For myself, me personally, as I've mentioned to you several times, neuroscience is not just an academic exercise. I went through depression. It was not something that I thought that I would ever go through, but yet I went through depression. I realized fairly late in my life that in my family, we also have a lot of family history of Alzheimer's disease. And even though I've been teaching about Alzheimer's disease for over 6 years, in terms of like knowing about Alzheimer's disease and then actually experiencing Alzheimer's disease and worrying about Alzheimer's disease every time I wake up in the morning and my memory's not so great, I'm always kind of wondering, like, you know, is my family history catching up to me? And I'll talk to you a little bit about that as we go on here today. But I don't want it to be merely an academic exercise for all of you in neuroscience. Neuroscience is very, very personal, and it should be something that you're investing in because you have a passion for our wonderful discipline. So I'm going to give you a really brief overview of Alzheimer's disease. We've talked about it before when we mentioned HM and we talked about memory. We're going to go through really quickly the gross pathology of Alzheimer's disease as well. You don't have to worry about things that are here over on the right. It's really just a cartoon in case you want to go and follow up on any of these things. You don't have to worry that it's ever going to show up on your final exam. In particular, we're going to be concentrating on two different aspects, and we're going to be looking at beta amyloid plaques, which you've heard about, these extracellular amyloid plaques, and then these intracellular neurofibrillary tangles. We're going to start looking at some treatment strategies. There is no treatment, as you all know, for Alzheimer's disease right now, but we're going to be taking a look, and you should know these for the final exam, different treatment strategies or putative treatment strategies that might be available. We'll also look very briefly at the genetics of Alzheimer's disease as well, and why some of the genetics and some of the predisposing factors actually link cholesterol processing as well as the late onset form of Alzheimer's disease. So why should you care? You should care because your prof may or may not have Alzheimer's disease, and it might or might not occur in your prof in the next little while. I don't know, to be quite honest. In my family, I've had one individual who had it in his 60s, and I had another individual, my uncle, who had it in his 50s. And I'm getting closer and closer to 50, and I never really know like what's sort of going on. But you should also be invested in Alzheimer's disease. As you likely know, there's going to be an increase in the prevalence of Alzheimer's disease. And by the time you're finished school, and by the time that you're out there either in research or by the time that you're out there practicing in the clinic, Alzheimer's disease is going to make an impact on you directly. It's going to impact someone in your family or one of your friends, or it's going to be impacting you directly. And it should not simply be an intellectual exercise for you. And as you can see, we're not really sure there are always these outliers. So it could be as high as 14 million, or it could be as low as 10 million. But overall, you can see that the trend is, because we're living longer and longer lives, that the incidence and prevalence of Alzheimer's disease is going to be increasing dramatically over the next few decades. And this is the decades where you will be at risk. So again, Alzheimer's disease, there are many, many different forms of dementia. There are things that are associated with cerebrovascular disease or vascular forms of dementia, but Alzheimer's disease taken overall accounts for roughly about 3/4 of all of the dementia cases that you typically encounter. So again, between 2000 and 2008, there was a robust 66% increase in the incidence of Alzheimer's disease and the incidence of Alzheimer's disease leading to death. And this actually far outstripped any of the other diseases. So heart disease actually went down during that period of time. Stroke causing death actually went down in that same period of time. So did prostate cancer, so did breast cancer, HIV-related deaths all went down. The one neurodegenerative condition that went up was Alzheimer's disease. And why is that? Because one, we have an increase in the incidence, but two, we have no effective therapy, and we have no way to actually make you get any better. Ashkin? It's entirely possible. So one, we're diagnosing it better. We simply have more people that are diagnosed with Alzheimer's disease, but this is also related to death. So those that are actually diagnosed with Alzheimer's disease are also dying at a higher rate, and it's gone up 66%. The new numbers over the last 8 years are going to be released soon, and I'm guessing that it's also going to be much higher than this 66% increase. So here you are as neuroscience students who are looking at trying to change the world with a lot of ideas and a lot of passion for your discipline. Maybe it'll be something that is personally investing for you, but we have this really bad statistic where Alzheimer's disease is going to be something that is going to go up in terms of related deaths, whereas every other discipline, whether it's oncology or whether it is something related to a communicable disease like HIV, everything else is going down, except in our discipline, it's continuing to go up. And again, Dr. Kalia will talk a little bit more about that with you in the upcoming lecture on Parkinson's disease as well. So Alzheimer's disease, if you're stereotyping it, is really this memory loss, but it's not just memory loss. For a variety of reasons that you should understand by the end of today's lecture, it's not just going to be memory loss, it's also going to be problems with attention. It's also going to be, because of the areas of the brain that get affected, difficulty with planning as well as solving problems, your cognition, in other words. As a result of all of these different factors, a lot of times what happens to these individuals is they often have confusion. So you'll wake up and you won't recognize your surroundings that you might have been exposed to for most of your life. And as a result of that, you have this spatial disorientation. So you suddenly can't remember having class here in room 1016, like, where am I? Like, who are these people around me? You can imagine how disorienting that would be. Because areas of the prefrontal cortex are affected and many of the cholinergic fibers are affected in Alzheimer's disease, it also results in very poor judgment or decreased levels of judgment making or decision making. There are also associated, as the disease progresses, changes in mood as well as in personality. Oftentimes there will be, as the disease progresses, problems with communication and difficulty in finding words. So whenever I'm up here and I'm kind of searching for a word and I'm kind of taking these long pauses, worry about me, because I'm worried about me, to be quite honest, because that never happened when I was younger, and it's only been fairly recently. It could be a lot of things. It could be sleep related. It could be related to dietary changes. It could be related to my mood or whatever else. But it is troubling that a lot of times I seem to have problems with finding certain words that I've been using in my lexicon previously. So we'll take a really brief look at the gross pathology, and we'll take a look in some more detail on the upcoming slides, but most of the neuropathology begins in the medial temporal lobe, and the hippocampus seems to be one target that is particularly affected, which is why memory gets affected in these individuals. Overall, the hippocampus tends to shrink. The ventricles, as the cortical tissue starts to atrophy and die off, the different types of the ventricles actually start to enlarge, and that's typical of Alzheimer's disease as well. And in severe Alzheimer's disease, you'll see very severe cortical shrinkage, as well as severely enlarged ventricles and severe shrinkage of the hippocampus. And again, there are two predisposing factors. You could have these amyloid plaques that are extracellular, and then you can also have these intracellular neurofibrillary tangles that we'll be talking about in a moment. Overall, this brain atrophy, the shrinkage of different neurons or the loss of these neurons begins in the medial temporal lobe. And again, the hippocampus is found in that medial temporal region. Then it moves into association cortices. Again, one of the reasons why you have a hard time finding words is the association cortex associated with verbal centers seems to get affected first. Sensory areas also get affected, motor areas also get affected in Alzheimer's disease. And again, many of the different neurodegenerative pathologies as they start to progress actually start to resemble one another. And you'll hear from Dr. Kalia that although it's a movement disorder, Parkinson's disease has many of the features that are associated with Alzheimer's as well. So the one thing, if you're going to take away from this slide, that might be a little bit new for you. One area of the brain that gets particularly affected in Alzheimer's disease is this nucleus basalis of Maynard. And this particular nucleus, which is found in the innominata striatum, it has a number of cholinergic projections to different areas of the cortex. And this nucleus basalis of Maynard, one of the reasons why it's so important is as it releases acetylcholine into different areas of the cortex, those areas of the cortex are responsible for things like sleep. Acetylcholine in those areas is really important for things like attention. So I told you that in Alzheimer's disease, there's a loss of the ability to pay attention to certain things, as well as for consciousness in certain areas related to projections to the thalamus. So overall, again, one of the things that you'll be surprised to hear about, sleep tends to be a really big factor in a lot of these neurodegenerative conditions. You'll hear about this several times before the end of the course. But before you see some of the signs and symptoms associated with neurodegeneration, as a subclinical feature of neurodegeneration, oftentimes you will have problems with sleep. And again, it seems to be a two-way street. If you are not sleeping, you might actually be predisposing yourself to developing these neurodegenerative conditions. And if you have these neurodegenerative conditions, it might also result in the lack of the ability to control your sleep as well. So if we took a look at the pathology in a little bit more detail, one of the things that you would see is that you would start losing these neurons. Not only would they start shrinking, you would also see the loss of neurons, not just atrophy, but neuronal loss. You would start to see the loss of synapses, and you would see as a result of the loss of synapses, the cholinergic projections from the nucleus basalis of Maynard would also be lost as well. And many of those areas would start losing their cholinergic projections. Overall, as the disease starts to progress, and there's been a lot of controversy as to which actually is more important and which precedes the other, the accumulation of extracellular beta amyloid plaques and the inclusions of these intracellular neurofibrillary tangles seem to be equally important. There has been some reports that have come out in 3D models using induced pluripotential cells that the extracellular beta amyloid plaques precede the generation of these intracellular neurofibrillary tangles, but that result hasn't been confirmed yet. And it's only a 3D in vitro model using induced pluripotential cells from an individual who has Alzheimer's disease. In addition to that, as neurons die, one of the things that happens is that the surrounding astroglial cells will start to form a glial scar, and that's typically observed as well. Just again, out of curiosity, because you're going to need to know these on different levels, how would I go back and mark histologically or using an antibody increased astrocytes or changes in astrocytes? What protein or antibody would I be targeting? GFAP, right, so perfect. GFAP is also a marker for what other cell type that we've looked at here in this course? Jessica? So the stem cells, or the adult stem cells that we looked at earlier, are also marked by GFAP. So glial cells like astrocytes, as well as GFAP found in those stem cells. There's also a robust immune response. And this immune response, there's always controversy as to whether or not this is good or the cause of this pathology that we see. And oftentimes the immune response in the brain, as you know, is a microglial response, and the microglial cells are the immune cells that we find within the brain. So I'm going to be concentrating on these two things, the extracellular beta amyloid plaques and the intracellular inclusions that result from neurofibrillary tangles. So if we take a look at the beta amyloid plaques, one of the things that you'll see is that they are found extracellularly. In culture, if you were to take these plaques that you observe here and you were to biochemically isolate them and age those plaques, and you were to dump them onto cultures of neurons, these plaques will actually induce cytotoxicity. And it's believed to induce cytotoxicity through a number of different actions. Different individuals have shown different types of things. It can disrupt ionic concentrations and might cause excitotoxicity. It can actually cause an aberrant action potential generation. But overall, these extracellular beta amyloid plaques induce cytotoxicity, at least in almost everyone's hands that have examined this. These plaques result from the cleavage of what's known as an amyloid precursor protein, or APP. And I'm going to use that short form of APP. You can use the short form APP on your final exam as well. And this APP is a normally occurring endogenous protein that we have on all of our cell membranes. And it seems to be a really important one. It's been implicated in a number of different normal cellular processes, and we have that. And it's a normal part of our physiology. This APP can actually be processed in two different pathways. There is a non-amyloid amyloidogenic pathway, which is caused by the cleavage resulting from alpha and gamma secretase activity. And then there is the one that actually forms the amyloid protein, which is known as the amyloidogenic pathway, which is caused by the cleavage, as the name suggests, by beta as well as gamma secretases. So this is the normal amyloid precursor protein. This is the cell membrane, and you can see there's a large N-terminal region that's extracellular, as well as a short C-terminal fragment. In the non-amyloidogenic pathway, and this might actually be a good pathway that your brain normally uses, this large amyloid precursor protein is cleaved by alpha secretases, which will cut at this area. And again, you don't need to worry about the exact position. And as a result of cutting in this area, it will produce this N-terminal fragment, the APP alpha, as well as this other intracellular peptide known as C83 peptide. And often you will see that in different publications. If you see APP alpha as well as C83, you know automatically it's the non-amyloid pathway that is caused by alpha secretase activity. The C83 intracellular peptide can further be cleaved by gamma secretase, and this gamma secretase is also found in this amyloidogenic pathway as well. And when it cleaves, it will cause the production of another peptide known as P3, as well as the A beta intracellular domain, often denoted in short form as AICD. Both of these, in turn, P3 as well as A beta intracellular domain, or AICD, are degraded. You have ways, proteases, that will naturally degrade these, and as a result, you don't tend to accumulate these, and these short forms are not thought to be neurotoxic. And as a control, when you put those on cortical neurons, these don't tend to cause cytotoxicity observed with beta amyloid. If you look at the other pathway, it's exactly the same the APP precursor protein. In the amyloidogenic pathway, we have activity of the beta secretase, and it cuts at a slightly different position than the alpha secretase, and it results, as a result, it produces a longer intracellular C-terminal fragment. So instead of being 83 amino acids long, this is now a 99 amino acid long peptide, which is why it's known as C99. So this intracellular peptide is the one that we're actually interested in. This C99 peptide, which begins with this beta secretase activity, is also processed by gamma secretase. But because beta secretase has cut this longer fragment, as a result, the gamma secretase, when it starts to cleave, it will actually produce the same A beta intracellular domain, but it actually produces a slightly different A beta peptide, which is either known as A beta 40 or A beta 42. So both isoforms, A beta 40 or A beta 42, are thought to be cytotoxic. Both of these are produced by the activity of gamma secretase. So I've taken you through really quickly the non-amyloidogenic, the normal pathway that we think is good for you, versus the amyloidogenic. And maybe it's really a balance between the two, because we do have all of these different enzymes. We have beta secretase, and why the beta secretase pathway and then the gamma secretase processing occurs more frequently in Alzheimer's disease is still a matter of great debate. So we have both of these pathways going at the same time. This pathway leads to non-toxic fragments. This pathway in red leads to toxic fragments. And the balance, when it shifts toward the red toxic amyloidogenic pathway, tends to be deleterious. And what the balance and the shift in balance is still, again, as I mentioned, a matter of debate. The intracellular inclusions, and I'll go through those really quickly, involve these neurofibrillary tangles. And these are composed of hyperphosphorylated tau proteins, and I'll go over tau really quickly in the next few diagrams. And normally tau is there to stabilize the microtubules that allow for axons to be formed and for axonal stability. And we need those axonal microtubules to allow both retrograde and anterograde transport from the synapses back to the cell body, and from the cell body out to the synapses in the anterograde fashion. So we talked about in the last lecture, pseudorabies virus being taken up retrogradely. It would make use of this microtubule system that I'm mentioning here. And the microtubule system, these microtubules get stabilized through the activity of tau. When when tau gets phosphorylated by different kinases, and it starts to get phosphorylated by these different kinases, it starts to lose its affinity for microtubules. So it's normally there sort of to keep the railroad tracks that represent microtubules in a stable fashion. But as it gets phosphorylated, it kind of comes off the microtubules, and the microtubules start to dissociate. As they start to dissociate, these will also start to disassemble. And as a result, the normal axonal integrity starts to get lost as well. And as a result of this, the neurons start to rapidly degenerate, and they rapidly start to die off as a result of these neurofibrillary inclusions. So normally we have these tau proteins that have sites for phosphorylation through different kinases, but they bind to microtubules represented by these blue and white spheres, and they act to stabilize them, to make sure that they're staying together. And these would represent the long tracks from the cell body, maybe out here on the left, to the different synaptic regions here on the right. And if they're not being held together by these tau adapters, they will start to disassemble, and the neuron starts to lose its integrity, and the axon starts to lose its integrity. So normally there are sites for phosphorylation, but as these kinases start to become overly active, it will start to hyperphosphorylate this tau. And as it starts to hyperphosphorylate, not only does it come off the microtubules, it will also start to aggregate with itself. So tau starts to form these large aggregates, which start to build up inside the cells. They begin as soluble tau aggregates, but as more and more of these tau soluble aggregates come together, they form these insoluble deposits that form these long fibrils. It will start looking like polymers that are found inside of the neurons. And again, these are intracellular inclusions, bless you, of these different tau fibrils, and along with these different neurofibrils that are associated with them. So we have both extracellular events going on as well as these intracellular events going on. As a result, the early neurobiologists and the early developmental geneticists took a look at all of the different biologies that were associated with these disruptions. So there are disruptions, for example, in amyloid precursor protein. Can we create a transgenic model that will recapitulate the amyloid precursor protein processing errors that we've just taken a look at? Could we further maybe look at another molecule known as presenilin or presenilin-1 and generate an animal model? And oftentimes you will see in the literature animal models that involve presenilin-1 and then all of these mutations that are associated with presenilin. By the way, that was found here at the University of Toronto in Dr. Peter St. George Hislop's lab, as well as APP in addition to this presenilin. Other individuals looked at the intracellular inclusions that we just looked at, hyperphosphorylation of tau, and they came up with different tau models or tauopathy models associated with aberrant phosphorylation as well. When they looked at these different Alzheimer's disease mouse models, either looking at APP processing or presenilin processing of APP or tau models, one of the things that has been somewhat of an issue in this field is that overall, if you were to take a look, much of these mouse models, whatever you're looking at, so for example, the APP model, APP23 mouse model that I highlighted for you, it's not highlighted, but it's the APP model. This APP23 represents an aberrant APP precursor molecule. If you were to take a look at just the histopathology, it actually recapitulates many, but not all of the features that are found in human Alzheimer's disease. So in terms of staining for the A beta plaque in a model, in a mouse transgenic model of amyloid precursor protein that gets aberrantly processed, we do find these amyloid beta plaques and that look similar but not identical to the human Alzheimer's disease plaques. But in terms of the progression of the different amyloid precursor changes that we see in the humans, you can see that it's not identical. So the human ones are shown to you on the right, and the mouse model, the overall time and the overall accumulation, you see a lot more of it in the cortical regions in the mouse, and you can see lots of A beta plaque deposition in the cortex. All these dots represent plaque deposition. You do get plaque deposition here in the hippocampus, but in terms of the progression in terms of humans, it doesn't recapitulate it exactly, and we'll talk about this as well. In terms of the, I think I actually mislabeled that for you. In terms of the neurofibrillary tangles, you'll actually see a difference in neurofibrillary tangles in blue. I have to apologize here on the right, and this represents neurofibrillary tangles. This represents the A beta plaques, and you can see again using different mouse models. Here is a mouse PR5 model, and this mouse PR5 model represents a tauopathy, that the accumulations actually are not identical between the different mouse models for Alzheimer's disease. You can see that if they were identical, the same region should be lighting up, but with the tauopathy version with the neurofibrillary tangles, in fact, we see more deposition in regions like the amygdala than we do in the cortex, etc. So they don't overlap 100%, and the two different mouse models don't equal human Alzheimer's disease as well. So this gets into, though, one of the things that I mentioned to you at the very beginning of the lecture, different treatment strategies that might be available. And these are different treatment strategies that many neurobiologists are taking a look at. So one way is to drive things into the non-amyloid pathway and to stimulate the alpha secretase. If we can get rid of as much of the processing of the amyloid precursor as possible by stimulating the alpha secretase, maybe that's one way to drive the balance. We could inhibit gamma secretase, because regardless of whether it's the 88 version or the 99 version, we want to be able to stop the gamma secretase activity of the intracellular peptide that gets generated. We could start doing passive immunization with anti-A beta antibodies to start eliminating the deposition of amyloid beta as soon as possible, or we might try using active immunization strategies where we immunize against A beta peptides. None of those have seemed, at least so far, to have been really effective. There have been clinical human trials using both strategies, passive as well as active immunization. And in humans, it doesn't seem to be very effective, at least so far. And again, here at U of T, we're part of a larger consortium, and many of you will have had courses with Dr. McLaurin, who's actually involved in the different clinical trials involving the immunization strategies against these A beta plaques. In terms of the neurofibrillary tangles, the inhibition of tau aggregation has always been sort of the target of this. A number of other ways to do this might be to inhibit those kinases that hyperphosphorylate tau. It might be to take the exact opposite approach. If you have hyperphosphorylation, how can you remove those phosphorylation residues? Maybe by activating different phosphatases. You could try using different chemical compounds that might increase microtubule stability that prevent the axonal disintegration, as well as potentially increasing tau clearance. There are many problems associated with this, and increasing tau clearance is hard because it's an intracellular protein, and it normally isn't something that gets outside of the neuron easily. So that's a potential problem in getting rid of these different forms of tau. So just in terms of the genetics of Alzheimer's disease, the sporadic forms of Alzheimer's disease cases are actually the major form of Alzheimer's disease. The familial forms of Alzheimer's disease, which occur at a younger age, only account for about 5% of all Alzheimer's disease forms of dementia. So familial Alzheimer's disease typically manifests itself a little bit earlier, somewhere between 40 to 50 years of age, or in that ballpark. In most cases of sporadic Alzheimer's disease, these individuals, we don't know the cause. It doesn't seem to be a genetically identified cause yet. It might just simply be because we haven't identified all of the different SNPs that might be associated with Alzheimer's disease. It might be something environmental and genetic that have to come together. We don't know, but at least in these sporadic instances, it typically hits after the age of 65. The symptoms, though, are identical. Whether you start off with familial or you have sporadic, the symptoms are identical in terms of memory loss, personality, concentration, inability to appropriately assign words. All of those things are found in both situations. In about 50% of that 5% of the familial Alzheimer's disease cases, we have actually identified, and I mentioned to you earlier, Dr. Peter St. George Hislop was the individual that actually found the presenilins. The genes are mutated in those individuals with these familial forms of Alzheimer's disease. In terms of why people develop Alzheimer's disease, in terms of the familial inherited forms, APP mutations, even though we have transgenic mice that have APP mutations and they don't fully recapitulate the human condition, these are relatively rare and only found in about 12% of familial Alzheimer's disease patients. Again, the average onset, if you have these forms of Alzheimer's disease, is in the early 50s. This is actually based on the observation that most Down syndrome patients also develop Alzheimer's disease after about the age of 40, and they also have an extra copy of chromosome 21 where we have this amyloid precursor protein. Most mutations alter the APP processing. So if you have a mutation in APP, even though it's really rare, it will actually increase the cleavage via the beta secretase pathway, but this results in the increase in the overall A beta 42 to 40 ratio, which means that the form that causes neurotoxicity builds up much faster. So remember, we always have a balance between the alpha secretase, beta secretase. If you have a mutation in APP, it will drive it toward the beta secretase pathway. So as a result of this, and as a result of forming this A beta peptide in higher mutation that's very commonly associated, 50% of all familial Alzheimer's disease, are the presenilins. The presenilins is actually not totally illogical in terms of why people might be looking at them or why they might be associated with Alzheimer's disease. Presenilins actually are comprised of gamma secretase. So if we were to take a look, this would represent the gamma secretase complex. Presenilin is one aspect of this gamma secretase complex. So we have either presenilin 1 or presenilin 2. We have another molecule known as nicastrin. We have another molecule known as APH, and then we have PEN 2 here in green. And this presenilin, if you have a mutation in presenilin, you will affect gamma secretase activity. And the presenilin component is the only one that actually has proteolytic activity. In other words, it's the presenilin that provides the gamma secretase with its ability to cause the further cleavage of the intracellular carboxy region of the APP peptide. So if you have a mutation in presenilin proteins, you have a lower age of onset of familial Alzheimer's disease. If you were to take a look, presenilin 1 mutations dominate any mutations in presenilins related to familial Alzheimer's disease. And again, if you have an increase in the mutation of presenilin 1 or presenilin 2, this increases the likelihood that you'll be generating the toxic forms of the A beta peptide. So again, genetics up until now can only explain a very small population, about 5% of Alzheimer's disease cases, and of those, about 2.5% are related to the presenilins that we just talked about. No gene has been found so far, and again, many individuals are looking for these, and as our genetic techniques become more and more sophisticated, I'm sure other genes will be found. But so far, no gene has been found directly responsible for this disease onset in the sporadic forms yet. Some genes are associated with a higher likelihood of developing AD taken as a whole. One of those risk factors, as you likely have heard in the popular media, involves this apolipoprotein E, or ApoE. And ApoE has been identified as the largest risk predictor. It means absolutely nothing, by the way, and I'll get into that in a few moments. It just means that it is a risk predictor for you developing late onset forms of Alzheimer's disease after the age of 65, but it is not a guarantee isoform versus the other that you will definitely have Alzheimer's disease. It's just a predictor only at this point in time. So in other words, you could have, as I'll show you in the next slide, we have 3 different variants of apolipoprotein E. So apolipoprotein E is normally produced by the microglia in your brain, as well as astrocytes within your brain. What is believed to be important for not just in the brain, but in other regions as well. One of the things that it does is it surrounds as well as solubilizes cholesterol that you might find in the brain, and we'll concentrate on the brain in these slides, allowing for its transport and removal from the brain. To date, there are 3 different alleles of ApoE that have been identified. And these 3 alleles involve the positions at 112 as well as 158. And if you have ApoE2, you have cysteine and cysteine at both 112 and 158 in terms of the amino acid residues. If you have So you either have Alzheimer's disease risk or you have a cardiovascular risk. Here, it's better to be average. It's better to be normal, in other words, where you have like ApoE3 status and you are normal for both cardiovascular as well as for um, risk of Alzheimer's disease. And again, as I mentioned to you earlier, why should it not be an academic exercise? In one of my other fourth-year courses in personalized medicine, because of Alzheimer's disease on my father's side, as well as risk of stroke on my mother's side. And so without even knowing that, because I couldn't ask my students to do it and we needed data, one of the things that I had done 6 years ago was to have myself genotyped. And unfortunately, without realizing that I carried this risk, I got genotyped, and one of the first pieces of data that came back was that I had the ApoE4 allele. And again, it didn't really bother me so much at the time because it's a risk predictor only. But again, knowing my family history, I might be a little bit more concerned now than I was earlier on. So why is ApoE all that important? ApoE has been shown to bind A beta, and different isoforms of ApoE will bind A beta and facilitate its clearance better than others. A decrease in ApoE levels overall will lead to an increase in the levels of amyloid deposition. And the ApoE4 isoform that I just mentioned to you actually correlates with the lowest levels of A beta clearance. So in other words, if you have ApoE4 status, you are more likely to have deposition of amyloid beta as an extracellular inclusion the older that you get, or you might develop it much more rapidly. Overall, it's the lipidation status. You actually have to lipidate ApoE to be able to activate it, to allow it to have this A beta clearance. And the lipidation and its subsequent association of ApoE with cholesterol is achieved by the activity of another sort of protein, which is known as the cholesterol transporter ABCA1, ATP binding cassette transporter 1. We need to have both of these, and I'll show you that in not this slide, but in the next slide. So overall, in terms of the role of ApoE and A beta clearance, it actually has a number of different effects that you should be aware of, and you should certainly be aware of these in terms of the final exam. So A beta gets cleared through the activity of different ApoE isoforms. So all ApoE isoforms do this. Some do this better than others, in other words. E2 would do this better than E4 would do this. But overall, one of the things that happens is that if you have ApoE and it's in this lipidated state, it will actually activate and turn on this enzyme that's found in the extracellular region known as insulin-degrading enzyme. An insulin-degrading enzyme will take the soluble form of A beta and actually make it degrade into soluble fibrils that can get cleared by the body. In addition to that, the soluble forms of A beta will bind to this lipidated ApoE, and it will actually cause it to be presented to these microglial cells where it can get degraded by this enzyme known as neprilysin, or NEP. And neprilysin will also, inside of the microglia, break down those soluble A beta fragments as they start to appear in a positive manner in these endosomal and lysosomal compartments. So they have a very important role in terms of clearance, both directly and indirectly through the microglial cells. But this also raises the point that there are targets that potentially that we can use in terms of drug therapy and treatment for the removal of these different proteins by knowing some of the physiology of the ApoEs. And I'll just cycle through this really quickly. The overall strategy is that we want to be able to decrease A beta plaque load. So if we have more and more accumulating A beta, it's not good for the cells. It might induce some cytotoxicity through a number of different mechanisms. The lower this A beta is, the better off you are. So ideally, you would have ApoE2, and you might want to start activating some of these other pathways. So if you look at the liver X receptor, or LXR, or RXR and PPAR pathways, if we start activating these, and you can activate these with a number of different drugs, if you activate these regions that are associated with gene activation, you can increase the levels of A beta C1, and you can also increase the levels of whatever ApoE that you have. And as a result of that, you will increase the ability for it to become lipidated, and you will be able to affect this particular pathway that we just talked about for clearance. And so you might increase the pinocytosis by microglia, and you might increase the degradation inside the microglia by that niprolysin, simply by increasing the activity of ABC1, which allows for this to become more and more lipidated. And this lipidated form of ApoE, again, turns on this insulin degrading enzyme, in addition to causing the presentation to the microglial cells. Hopefully that makes sense. So if you can target drugs that will act on this portion of the promoters that are responsive, like liver X receptor or RXR or PPAR alpha and PPAR gamma, if you can activate these regions, then you can turn on the expression of ABC1 or ApoE. Yep. These soluble forms, they can get cleared normally. Yeah, there are a number of other routes that this can actually get cleared. I'm not talking about other physiological factors. So in addition to all of these factors, one of the things that seems to be really important in this clearance, so even if you push things in this pathway, which is why I keep talking to all of you about getting a good night's sleep, one of the things that seems to be really important in this pathway is that when you're asleep, the clearance of many of these breakdown products seems to occur at the highest levels when you're sleeping. It doesn't occur when you tend to be awake. And that was a series of papers that came out last year in Nature and Science using the overall changes in vasculature that occurs in the awake and sleep animals and the overall accumulation or clearance of amyloid beta. And if you're really interested, email me. I can send you the paper. It's a technically dense paper involving inulin clearance as well as transgenic models. But it was a staggering paper that suggested that getting a good night's sleep is overall really important for your brain health in clearing off metabolites, not just related to amyloid beta clearance, because all of you are generating all of these different fragments now. It doesn't occur late in your late in age. It's occurring right now in your brains, and your ability to be able to clear these and not form these different plaques is really important. And the earlier you can do it and the longer you can sustain it, the more important it is for your brain health. Okay, so if you can target different drugs to activate these pathways, it might be beneficial because you'll be activating both ABC1, which causes lipidation, as well as creating more of this free ApoE. So you have so many of these different drugs that have been found so far in terms of Alzheimer's disease therapeutics, many of the drugs have some serious side effects that are associated with it. Many of the therapies, and again, there was great news in the early to mid 1990s, where in animal models that I showed you, so there were a number of transgenics. You could look at APP models, you can look at tau models, you can look at presenilin models. As soon as those animal models were created, people started generating different therapies. It worked fantastic in animal models, but it didn't seem to be very effective in human trials. Same thing with the antibody trials that I mentioned to you earlier. So plaque clearance in Alzheimer's disease might be achievable at some point, but it's not thought to be reversible. So once you've crossed a certain threshold, even taking these drugs, it might be too late. Again, which is why in neuroscience, you try to be as multidisciplinary as possible. And we talked about stem cells, and certainly stem cells might replace the neurons that are already lost, or it might be too late to allow this to occur. And then how this is all going to be overcome, to be honest, we have really no clear idea. That's really in your hands. And one of the things that I started off with at the very beginning, it's in your generation as young neuroscientists or as clinicians, if you're out there in the healthcare field, you're going to be observing more and more cases of Alzheimer's disease and associated dementias, and you're going to be researching it more. You have all of these sophisticated techniques, and it's the ideas that are ultimately going to win the day. And knowing a little bit about the biology now, you know some of the things that stand in the way in terms of hurdles to developing these different types of treatment strategies. So do you have any questions before we wrap things up? It's a downer, isn't it, for Tuesday morning? That's okay. I have tremendous faith in this class to be able to come up with great ideas and potential therapies. You're one of the most bright bunches of students I've ever seen, so I'm sure that you guys have lots of ideas and that you will be able to at least think outside the box in terms of the therapies. So again, one last thing before you go. If you need any comments back on your upcoming assignment, feel free to share as a Google Doc. Do not email it to me. Share it as a Google Doc. If I get it before noon tomorrow, I will send back comments to you about how you might improve it or if you are doing just fine. Okay? Have a great week.